Hello. Today I'm going to talk about finding transcription factor binding sites or operator in bacterial genomes. This is important because we have many thousands of bacterial genomes in GenBank, but most of them have just an automated annotation, and such an annotation usually has just genes and their products and doesn't have promoters, transcription factor binding sites, and terminators annotated. So we have tried to make our own solution to this problem. And what you see here is a screenshot from our application written a couple of years ago. This application is built around a genomic browser. Can show the annotation from GenBank files, can show RNA-seq data, and can, of course, find and annotate promoters, operators, and terminators. The basic idea behind this program was to use existing information from transcription factor binding site databases like RegulonDB, CollectF, and RegPrecise, and transfer this information to uncharacterized genomes. What can you achieve with such an approach? This is again an example from our paper. We have found 74 transcription factors autologous to known characterized transcription factors from other organisms. I have found about 1,000 of operators for these factors. And this was sufficient to explain a transcription regulation of about 40% of transcription units. There is a problem with this approach because all this is based on the assumption that homology between transcription factors is sufficient to assume recognition of similar operators. And that assumption is not always true. So we have tried to find a better solution and to find a criterion that is more strict, more formal for the transfer of existing information to uncharacterized genomes. And the answer to those two questions is actually yes. We have this information all the time. There are a lot of structures in protein data bank of transcription factors with their operators. And you can actually see the contacts between amino acid residues of transcription factor DNA binding domain with the DNA bases. And it is this contact that allow transcription factor to read the information from the operator. Fortunately, the structure is always, in most cases, is the same in bacterial transcription factors. It's helix-stem helix, and it is this recognition helix that is responsible for most of the contacts. And the factors are symmetric, so the operator is symmetric too. It's either palindrome, as in this case, or direct repeat if we have different packaging of identical subunits of transcription factor. If you take a family of transcription factors and align the DNA binding domains and look where the amino acid residues are located, those that contact the operator bases, you see an interesting picture. You have blocks of these amino acids separated by residues that are not involved. What you see here is actually three consecutive turns of recognition helix: the first turn, second turn, the third turn. So you can record the positions of the contacting residues, and you can actually write down amino acid residues that sit in these positions. And this way you get sort of a fingerprint that uniquely identifies the pair of transcription factor and its operator. It's obvious that transcription factors with the same residues here recognize the same operators. So we can call this amino acid residues critical residues, and we call this fingerprint critical residue tag or CR tag. And again, it's a unique identifier of TF and operator pair. The idea to use CR tags for finding operators was suggested about 10 years ago by Sahota and Stormer, but for some reason it hasn't been used by bench scientists, and we somehow actually missed this paper when we were developing our sigmoid application. So once we came across this paper, we tried to add this idea to our program, which proved to be quite difficult because the situation have changed during these 10 years. And the amounts of information increased, and you have to handle all this information somehow. So this is the block scheme of our approach, our method. It's based on two assumptions. The first one I have already mentioned, transcription factors with the same CR tag recognize the same operators, and it's true all the time. And the second assumption is that transcription factors are likely to have at least one operator near their own gene. That assumption holds true in most cases, but there are exceptions to this rule. So how does it work? First, we find transcription factors that belong to the same family. We use hidden Markov models from PFAM database to scan proteome of organism under study. Then we have a list of transcription factors. And for each transcription factor from that list, we find homologous proteins in protein databases that have the identical CR tag. That's actually a critical stage because it's time consuming, and we have replaced database age with a simple lookup tables that have CR tags matched to IDs of proteins with the same CR tag. So instead of doing time consuming database age, we just take identifiers from the table. And there is a poster that has more detail on this approach and additional benefits of it, redundancy reduction by Pavel Vichek in this conference. So I'm not going into further detail here. So the next stage of our pipeline is extraction of regulatory regions of those steps that we find at the first stage. This is a dedicated problem because you have to correctly find these regions, and we have two examples here. In this example, which is the simplest, we have transcription factor at the end of its own operon and two binding sites in front of that operon. So basically, we just look for the start of this operon and extract the sequence between the first codon and the end of the previous reading frame. The second example is a bit more difficult and more typical. In that case, again, we have one binding site in front of transcription factor on operon, but we have three more sites in front of the other operon, single gene operon in that case. So in order not to miss any information, we actually extract three regulatory regions. This one, the one from operon to the right, and the one from the operon to the left of the transcription factor. So we combine those three regulatory regions and get a sort of super promoter. And then we have to collect at least 10 of such super promoters, preferably more. And then we feed these DNA fragments to motif finding programs, which will always find some motifs. Actually, we run a motif finding program, MIM in this case, in two modes. One looks for a single motif, another one looks for any number of motifs. And you can see that we get quite different motifs from these two modes of the same program. So basically, we have to choose which of the 10 motifs is the correct one for the given transcription factor. And that slide illustrates a complicated situation when we have three transcription factors surrounding one block of cell biosis utilization genes. And in that case, it is this motif that is recognized by that transcription factor from the GenChar family that is responsible for the control of those genes. How can you determine this? We do this by comparing every motif to a library of well-characterized transcription factors. The TomTom program, which is from the MIMSUIT package, is built in our pipeline, and it does a pairwise comparison of the motif we find at the previous stage to the library motifs. It gives you the alignment, it gives you the statistics that shows you how significant it is. And in most cases, this is sufficient to decide on the correct motif, because the motifs from the same family have similarities, and usually just one motif that matches motifs from the same family. In more difficult cases, we have to resort to a slightly different approach and use clustering of motifs of our motif with the motifs from the same family, and look for CR tags of the similar motifs. And the idea here is that very similar motifs should have very similar CR tag. And if this is not the case, then something have probably gone wrong. And in that case, everything is fine. Similar motifs have similar CR tags. So because of those complications, we could not build a completely automated pipeline. And the final stage, the actual decision on the correct motif, is left to the user, to human user. So our pipeline is actually semi-automated. So what can you achieve with such an approach? This is again an example of a plant pathogenic bacterium. In that case, we were able to characterize 164 transcription factors of about 250 that are present in this bacterium. We could identify correct motifs for 39 or 42 non-transcription factors, which is quite good, 93%. And for uncharacterized transcription factors, we get typical for the family motifs in 3/4 of the cases. But the typical for the family is probably not quite convincing. And of course, you have to experimentally test all these motifs. And it is basically just a testable hypothesis that you can get from this approach. Is there a more reliable way? Yes, you can do a bit better if you have a large library of CR tag trusted operator profiles. As I have already mentioned, if CR tags of transcription factors match, then their motifs should match as well. You can basically use the same motif for different bacteria if the CR tags are the same. So, but where can you get such motifs? There are two databases with these motifs, but first we look at this slide which shows you what you get in the end. So if you apply the CR tag labeled library, you can get quite good results actually. In that case, with one-power family, with 9 profiles based on Escherichia coli data, you have 3 genomes with identical CR tags for each of the 9 transcription factors, 3 more genomes with just 1 protein having different CR tag, and 1 more genome with 2 proteins with difference. All the rest are the same, so you basically just transfer this information directly. For other less conserved TF families, this approach is less efficient, but still you have about 50% of profiles that you can use. All this works well within the same taxonomic order. So in that case, in Enterobacterialis. But where do you get the trusted profiles? The best source is probably Regulon DB database of Escherichia coli motifs. But the problem with it is that more than half of the transcription factors don't have motif. And there are 2 main reasons behind this. The first one is that most of them have just one or two binding sites. And another reason is misalignment of operators or imprecise definition of the operator sequences. And in all those cases, you have no motive. So how can we solve this? We just apply our de novo operator finding approach. And if such a motive finds the sites mentioned in the Regulon DB, then we consider this motive a good one to recognize these operators and consider it to be based on experimental data. Another approach is to use a reg-precise database, but the problem with that database, which is the largest, is that it has little experimental support and tends to combine the data from transcription factors with different CR tags. In that case, you can see different CR tags due to different genomes used, but in some cases, the data for paralogous regulators are combined, present in the same genome. And here is the illustration that shows you that the data from the previous slide are combined incorrectly. Again, this is an analysis of Pictobacterium genome, and we have 4 paralogous transcription factors from LACA I family, recognizing similar motive, similar but different motives. And with our approach, we can assign correct motive to each transcription factor, find correct targets for each transcription factor, 1 target in that case, 2 targets in different places for this transcription factor, again, 2 targets for this transcription factor, and ironically, no targets for this one. And in that case, similar genes are controlled by a different family transcription factor. So I have a couple of more practical examples from our experimental work. This is data from our transcriptomic experiment. And in that case, for this plant pathogen, we have about half of transcription factors change in expression level in host plant. So we are looking what these transcription factors control. So we basically take the transcription factors that have the most differences between in vitro and in plantar conditions and look for the motives and can actually find some interesting things. In that particular case, this is LISA regulator, and this is its motive. The only target is CSRA regulator, and it's global regulator of virulence factors. We have overexpressed this gene and show that it indeed does affect expression of pictate lyases and cellulases. Another example here from our another transcriptomic experiment, the 4P transcription factor controls arabinose utilization genes, but we can find binding sites in front of just one of those genes. So we have looked for control of other transcription factors, found this one, characterized its motive here, found binding site for this transcription factor in front of those 2 arabinose related operons. And there is the RRC transcription factor here, which controls the last one operon. So we have a sort of transcription cascade here. First transcription factor, second, and the third one. And so our approach helps to decode the regulation in that case. So final notes, our approach is universal, can be applied to any bacterial genome. Complete genome-wide scan finds regulators for the majority of transcription units. It finds unexpected regulators for many operons, and method efficiency should improve as the size of reference library increases. And our application is freely available, and please come to that site and try it. Thank you for your attention, and I'd like to thank my colleagues that helped me to do this.